Here's a quick explanation about how DNA stores information. James Watson and Francis Crick discovered the structure of DNA in 1953. Their work revealed the remarkable way that DNA stores information. They found that DNA, its full name is deoxyribonucleic acid, stores information using a simple code. The code has 4 letters and involves a cool feature called complementary base pairing. So, how does complementary base pairing work? If we flatten out a DNA helix, it would resemble a ladder. In DNA, each step in the ladder is made of 2 pieces. These pieces are called bases. DNA has 4 bases, named adenine, cytosine, guanine, and thymine. We abbreviate them as A, C, G, and T. These 4 letters are the DNA code. A key part of Watson and Crick's discovery was that these bases form pairs in a very specific way. A pairs only with T, and C pairs only with G. That is, A and T complement one another, and C and G complement one another. This also means that C and G cannot pair with A or T. By the way, an easy way to remember which bases pair with which is to write the letters in alphabetical order, then below that, write the letters in reverse alphabetical order. So, what does all this mean for a cell? When a cell prepares to divide, it replicates its DNA so that both daughter cells have exactly the same genetic information. Complementary base pairing means cells can replicate their DNA quickly and efficiently. During DNA replication, the double helix separates down the middle where the pairs of bases join. This exposes the sequences of A's, C's, G's, and T's on each side. Then, groups of enzymes come along and add complementary bases one after another along the entire length of both DNA strands. That is, gene after gene along the entire length of the chromosome. In the end, there are 2 identical chromosomes encoded with the same genetic information. Complementary base pairing also plays a critical role when cells make proteins. During protein synthesis, the DNA, that is the gene that codes for the needed protein, opens up. This exposes the sequence of bases in that gene. The gene is copied in the form of RNA using complementary base pairing, but with one important difference. RNA has no T. Instead of the base thymine, it uses a base called uracil. So when a gene is copied during protein synthesis, for every T in the DNA, it is matched with a U in the messenger RNA. You can learn more about protein synthesis in the video How Genes Express Themselves. Scientists also use complementary base pairing to help understand how cancer happens. For example, scientists also use complementary base pairing to help understand how cancer happens. For example, if they have one strand of DNA or just part of a DNA strand, they can use complementary base pairing to tell them the sequence of bases on the opposite strand. Gene sequencing technology reveals the lineup of bases in DNA and in messenger RNA. Scientists can then use complementary base pairing to identify the gene that produced that messenger RNA. Once scientists know the sequence of amino acids in a protein, they can work backwards and decipher the sequence of bases in the messenger RNA for the protein and then the gene that codes for that protein. It also allows scientists to compare genes from cancer cells and healthy cells from a patient. This helps them learn more about why cancer happens and how tumors grow. Watson and Crick's work explained human heredity and opened a new field of molecular biology. It also earned them a Nobel Prize in 1962. Pretty nifty discovery, huh? If you'd like to learn more about the discovery of DNA, read the book The Double Helix by James D. Watson, shown here in an annotated and illustrated edition.